Hello, everyone. My name is Jeff Kordower, and I'm the founding director of the ASU Banner Neurodegenerative Disease Research Center at Arizona State University. I'm excited to be the moderator of today's session, and we have an outstanding lineup of world-renowned scientists who are all passionate about studying multiple system atrophy. We have Dr. Lucy Norcliffe-Kaufmann, we have Dr. Patrik Brundin, we have Dr. Ronald Melki, we have Dr. Sara Holec, Dr. Michael Henderson, Dr. Wouter Peelaerts, Dr. Amber Van Leer, and Dr. Ziv Gan-Or, and Dr. Sandra Bettenhausen. And we'll go around and ask questions. But before, although all of them have given talks at this conference, Dr. Van Leer has not, but she's been invited here because she's got some very exciting news to share with us with regards to a clinical trial that she's involved with MSA. So Dr. Van Leer, would you just inform the audience here about what's going on with S-Bio?
So thank you for having an opportunity here to let everyone know about the study. Just for clarity, it has not started yet, but we're looking to open this up very, very soon. And so I wanted to make sure it was brought to the attention of the MSA community. So in short, we're using gene therapy as a potential treatment for MSA. I'm not sure if anyone here was able to attend the talk yesterday, but we're using basically brain surgery to help us deliver this therapy directly to a part of the brain that's affected by MSA. So we're going right to the putamen. We're using a drug, it's called AAV2 GDNF. So this is a type of gene therapy that we have a lot of experience with Parkinson's disease. And so we have good reason to think it may be a good starting place, at least, for looking at MSA. It turns out that when we look at tissue from patients who've passed away and we look right in their putamen, they have very low levels of GDNF. And so this is an opportunity for us to directly replace GDNF in this part of the brain that's really insufficient. We have a lot of reason to think that based off of what we know from Parkinson's disease and even some animal models of MSA looking at GDNF replacement, we have some reason to think this might be successful. So just wanted to bring some of that to everyone's attention today, and happy to answer more questions about it. I think there's a few that are coming through already.
So you mentioned that it's brain surgery, and I once got assaulted verbally by my dear friend Andres Lozano that I called it minor brain surgery. There's no minor brain surgery, but there's an evolution now about using focused ultrasound to, without surgery, opening up the blood-brain barrier and delivering proteins that normally don't get into the brain. Is that something that S-Bio might be considering down the road?
Yeah, it's definitely being looked at. I think that it's still not perfected yet, let's say. I think there's still concerns about getting enough of your actual gene therapy product and making sure it's going to the places where you want it to go. One of the big benefits of doing the delivery that we're doing is we actually are kind of taking advantage of the network that's already been established, the nigrostriatal network or the striatonigral network. So we're taking advantage of that to really not just affect the putamenal cells that are there, but also the entire striatonigral trajectory as well. And it's harder, I think, to get enough of a vector in the right spot in order to achieve that same transduction if we're using the focused ultrasound. But maybe there are ways to enhance this to get this better. There's other ways that we're also exploring to get around the direct delivery of gene therapy, period. But that's also still in its infancy. But it's something that's still being explored. So we could potentially do IV delivery and not have to mess around with direct delivery at all.
Thank you very much. And this is exclusively for MSAP versus MSAC?
Yeah, so there's rarely you're going to find a purely, MSAP patient. Like we understand that, but we have reason to think that with, again, our experience with Parkinson's disease and its delivery right to the putamen, that we have evidence to suggest that this is helpful really for the motor symptoms more than anything. And building off of that experience with Parkinson's, we have reason to think that this approach at least is better for folks who have more Parkinsonian type of symptoms. But this is still early days. If there are cellular features, that's not an issue. We just want to make sure that there are Parkinsonian features there because we think that's really what's going to be benefited the most.
Thank you very much. Dr. Melki. one of the things that's impressed me when I've seen patients with MSA is just how aggressive it is. In most cases, there are some where it's not, but in most cases, it's just incredibly aggressive. Can you explain to us why you think that MSA is far more aggressive than Parkinson's disease in terms of its progression?
Well, I'm convinced. Thank you for this very nice question. In fact, we don't know, but we can hypothesize. So what I can say is that the structure of MSA fibrils is different from the structure of PD fibrils. So you can imagine that the proteins and the cells that these fibrils interact with when they propagate are different. Some potentially are the same, but also some are different. And it's, again, the surfaces of these fibrils that are important. The surfaces are not only the lateral surfaces, of course, but the ends as well. And it's through the ends that these fibrils grow and recruit endogenous alpha-synuclein. So I think that they are very aggressive because of this, because they have different surfaces, they target different cells, but also they grow differently. They interact with proteins within the cells with maybe different proteins, and some are very important proteins. And what you need to understand is that when a fibril interact with a protein, it sequesters the protein. So in fact, we are sort of mimicking a loss of function. And it is through losses of function, multiple losses of function, that we have pathology.
Thank you. Dr. Peelaerts, as we were talking, there's some controversy with regards to where the GCI, where the synuclein comes from for the GCIs, the glial cell inclusions. Early studies showed a lack of mRNA in the oligos, and later studies are showing that there can be mRNA. Could you expound upon that and clarify that for us?
Yes, I also find that a very interesting and intriguing question, because in order to really have pathology in these oligodendrocytes and to have MSA to develop and progress, I also believe there has to be expression of alpha-synuclein protein within these cells. And so these building blocks, alpha-synuclein monomers, I think, therefore has to be present within the oligodendrocytes. And so some people have come up with a few ideas or a few hypotheses as to how these oligodendrocytes are expressing alpha-synuclein. Now, one way how the synuclein might end up in the oligodendrocyte is by transfer from neurons to oligodendrocytes, and that might lead to a buildup of the protein itself within the cells. Another idea or another hypothesis that's being discussed is that some sort of trigger might actually trigger the expression of alpha-synuclein within oligodendrocytes, even in a transient manner. What is, I think, more established is that in multiple system atrophy, these oligodendrocytes have an impairment in the degradation machinery, that they cannot clear alpha-synuclein as efficiently as, for instance, normal people do. And that might, for instance, also allow a buildup of alpha-synuclein and then those toxic species that I think Ronald has talked about so much. But how this exactly happens and where the synuclein is actually coming from in these oligodendrocytes is not very well known.
So let me follow up. You're a world leader in the area of animal models of MSA. And so if you had a therapy that you thought would be helpful in patients with MSA, which model would you choose? And hopefully you'll say the model that I work.
I'm sorry, come again?
Go ahead. Just which model would you choose?
I would certainly choose a model where there's expression of alpha-synuclein in these oligodendrocytes, and then so leading to the accumulation of the protein. And this is something that we've also seen, and this is very consistent. Once you overexpress or express the protein in transgenic models of multiple system atrophy, then this will automatically lead to deficits, demyelination, even several of the motor symptoms that we see in people with multiple system atrophy. So even though we don't exactly know how this disease starts and where even the protein is coming from, I think it's a valuable model because it can still tell us much about the disease, and it can still tell us and give us good information about how to treat and target this disease. So I think for now, this is probably one of our best models so far. But I also would note and mention that we have to better understand how this disease comes about, how it develops, and by doing so, hopefully developing animal models that mimic the disease much better than they do today.
Thank you. Dr. Gan-Or, could you explain what epigenetics is, and do they play a role in MSA?
Sure, I can. I think maybe there are also some other people here who can answer this question just as good as me. Epigenetics is a general name for a mechanism by which external stimuli can control or can guide how DNA is being transcribed and translated in the cells. In other words, epigenetics is a way of the cell to control whether a certain gene or a certain protein will be expressed and to which level. Now that can be, epigenetics can be affected by many things. It can be affected by medications, by every environmental exposure that we have can affect our epigenetics. And unfortunately, we don't know much about how epigenetics affect MSA and what role exactly does it have. And it's quite complicated to study because of the exact same reason that I mentioned, because there are so many things that can affect epigenetics. And if you test it in different people, or even if you test it in the same people, over time you will see changes in the same individuals. So it's hard to say what is the, currently it's hard to say what is the contribution of epigenetics to MSA.
Okay, thank you. Dr. Brundin, there are different forms of MSA. A questioner asked, why don't they use MSA-A anymore? And could you compare and contrast the forms of MSA that are seen in Japan versus North America and Europe?
So I must say, I almost feel like I'm back in school. I'm nervously waiting for my question. And I got a tricky one. It's true, there are different forms of MSA, and they are based on the anatomical distribution of the pathology and as a consequence, the types of symptoms. And the most commonly occurring ones are the MSAP, where the P stands for Parkinsonism, where the basal ganglia are affected, and one sees symptoms that are, of course, are similar to Parkinson's disease, slowness of movement, rigidity, and so forth. The other major form is MSA-C, where the C stands for cerebellum, where the pathways that are connecting the cerebellum to the rest of the nervous system are also affected. And they have slightly different types of symptoms, more related to coordination of ongoing movements. And the question is, I think one should ask the question, should we even call these diseases the same thing? It might be confusing us a little bit, because even though they both have the aggregates that we just talked about in oligodendrocytes as a major feature, they are so different in their distribution of pathology, etc. So they might actually be two totally different entities. And if you think of what I spoke about in my presentation, one would then suggest that the MSAP cases would be completely distinct from MSAC in terms of their origin. So a urinary tract infection would most likely not have anything to do with MSAP, because the distribution of the pathology is so different. It has no connection to the urinary tract. And I think we would probably learn a lot more if we decided in the genetic studies and all the epidemiology studies, all the animal model studies, all the protein aggregate types of studies, to distinguish between these two forms, these major clusters straight away. And as you hinted, there's slightly different frequencies of these forms in Asia and in the Western Hemisphere.
Thank you. Dr. Henderson, some MSA patients have dementia. Do you think that it's the alpha-synuclein aggregates that are involved in that symptomatology, or do you think there's co-pathologies such as tau or amyloid that are participating in the cognitive decline in some MSA patients?
Yeah, that's a great question. So for Parkinson's disease, for example, while we think of Lewy bodies as the primary pathology, we know that as disease progresses, these patients can accumulate additional pathologies, such as the tau tangles and amyloid plaques that you would see in Alzheimer's disease, and these have some correlation with the progression of these patients to dementia. So does this happen in MSA? I'm not as familiar with the literature. I think we've just not done as many studies on what the co-pathologies that may modify MSA are, but certainly you could also imagine that if, for example, you get oligodendrocyte, these glial cytoplasmic inclusions in areas that control cognitive function, then that itself could also lead to cognitive impairment as well. So great question. I think it's possible there's modifying co-pathologies, but probably more work needs to be done there.
Thank you. Dr. Holec, is there any way to bust the fold of the proteins or straighten them out?
In the laboratory, there is. We commonly use a chemical, or there's multiple chemicals we can use to kind of straighten out the protein, and that's one of the ways that we can differentiate between different strains of protein-caused diseases. In patients, however, in terms of thinking, can we do this in a patient? That's a lot more tricky, because anything we would use to unfold a protein in the laboratory would unfold all of the proteins. And so naturally, that's probably not something we could ever use as a therapeutic, but it's very useful for researchers to understand how this folding can then lead to disease. So that's something that we work on. And yes, we can bust the fold in the laboratory.
Well, you could also prevent the fold from ever occurring. You could use things like nanobodies or immunotherapies, and those have clinical utility going down the road.
Yes.
Okay, Dr. Norcliffe-Kaufmann, how long are natural history studies, and are they normally a precursor to interventional studies?
So the natural history studies usually run for a good number of years. They can involve an intense, they can involve an intense period of being followed, and then usually a period where patients are called in or do remote visits to be followed. It depends on how the study is set up. The current one, the natural history study that a lot of us have participated in, has been going on for almost a decade. So quite often those studies, because they're such a wealth of information, they keep adding and adding more years as they follow patients through. But if you're considering participating in a natural history study, you should think of it kind of in terms of like it's going to follow you, throughout your lifespan and track their clinical stages, because one of the most important things about those studies is they capture all phases of disease, including, the later stages. And in terms of the second question about whether they're always a precursor for an interventional study, I think the way to think about it is that these two are complementary, and that the information generated during a natural history study supports multiple interventional clinical trials in terms of knowledge and understanding. So we'll understand better how those diseases, how MSA evolves, how to track it over time, and they can be used to develop or refine new scales to measure progression. So they really are sort of complementary, and one doesn't necessarily, and the interventional trials don't necessarily follow on from that, but there's a lot of interplay between the two that they can help support clinical trials in multiple different ways. I hope that answers the question. I know we had a lot of questions about who can participate in this and whether you need a caregiver. That was one question that came in from the audience, but I just want to emphasize that these studies are available to anyone with an MSA. It doesn't matter on the disease stage, and you can participate with or without a caregiver.
And can Canadians get involved with Track MSA?
with track MSA not directly involved because it's being currently carried out at 2 clinical sites because there's a need within the natural history studies to keep things in this early stage of very intense follow-up at sites where we can standardize the measuring techniques and know if we're tracking something that's worthwhile or tracking something that doesn't follow the disease progression. But having said that, the natural history studies are very collaborative, right? So, keep checking back to see if like other sites have been added, and generally the goal is to put together as many sites to contribute as possible because there's a wealth in having more information.
Okay, thank you very much. Dr. Gan-Or, there's someone in the audience who's interested in RBD. And is RBD a prodromal symptom of MSA? And if so, is there something that someone can do to prevent the evolution from RBD to PD and MSA?
Thank you for the question. So RBD can progress into Parkinson's disease, dementia with Lewy bodies, or multiple system atrophy, but only a small minority of people with RBD, about 5% of them, will develop multiple system atrophy. It's very common in multiple system atrophy, but some people develop it after MSA starts. In terms of if there's anything that can be done about it, unfortunately, there is no way currently to slow down the disease progress, as far as we know, maybe except for doing some physical activity. But and which is true for Parkinson's disease. But for MSA, we don't have studies specifically about that. And as I said, there's no specific recommendation that I can give.
Okay, thank you. Dr. Melki.
So sorry, could I just add that? Because
sure,
I saw just below that question, there was another BD question, not RBD, but IBD. And IBD is inflammatory bowel disease, as opposed to REM sleep behavior disorder, totally different conditions, very similar abbreviations. But there is a link between IBD and multiple system atrophy, and Dr. Gan-Or apparently mentioned this in his talk. I was not able to listen right then, but it's a paper that came out earlier this year that suggests there's overlap in the genetics of inflammatory bowel disease and multiple system atrophy, which is very interesting in many ways. One could imagine that there's a common cause that can manifest in two. One could also imagine that inflammatory bowel disease is a risk factor for MSA, because when we talked about the urinary bladder, we only talk about the urinary bladder in our experiments because that's what we've been experimenting with. But the nerves that innervate the urinary bladder overlap with the nerves that innervate the lowest part of the intestine, so the rectum. And proctitis, inflammation of the lowest part of the intestine, is very common in both ulcerative colitis and Crohn's disease. So no one could speculate, and this is just speculation, but inflammation in that region would trigger synucleinopathy in the nerve endings that then would lead to the spinal cord segments 2 to 4 of the sacral spinal cord. And this could be the common link. Totally speculation, but it's an intriguing speculation.
Thank you, Dr. Brundin. Dr. Melki, could you address the issue of mitochondrial dysfunction in MSA? And is there something that we could do to correct that?
Well, unfortunately, this is an important question again. And unfortunately, what we cannot really much act on mitochondria. What we need to do is act ahead of having the fibrils getting inside our cells and multiply and affecting the mitochondrial integrity. So we really need to target these aggregates. I mean, we have now evidence that these aggregates form a long time before they are found in the brain, in the periphery. And they can be used, of course, to diagnose the disease to some extent. But I think we need really to target them with antibodies, as you mentioned at one point, with other ligands. And we are trying to develop such ligands before they get inside our cells and cause, the damages they cause.
Thank you. Dr. Henderson, are there other proteins involved in MSA besides alpha-synuclein in terms of pathology?
Yeah, that's a good question too. So alpha-synuclein is what we use to kind of mark which neurons, or in the case of MSA, which oligodendrocytes are affected by pathology. But certainly they're not the only proteins that are present in pathology. There are a lot of other proteins that become incorporated in these inclusions. There's kinases, there's organelles. And so what these inclusions may be doing is disrupting some cellular functions related to these other proteins. And so what's interesting about MSA that's different than Parkinson's disease is the cell type. And so Wouter briefly mentioned this study that found that if you have pathology that begins in oligodendrocytes, it's much more potent. And that what that study had proposed was that it's actually the cellular environment of oligodendrocytes that somehow modifies that pathology in a way that makes it more pathogenic. So that's a long answer to say, yes, there are other proteins involved. We don't know the identity of all those, but certainly in differentiating PD and MSA, there may be really important different cell type environments that modify that pathology in the way that it acts in the brain.
Thank you. Dr. Wouter, any way to get these cells that are lost back?
Ooh, that's an interesting question. That's maybe a question that I could also ask Patrik maybe in a bit, but well, and of course you as well, Jeff. But so there is of course potentially stem cell therapy there. I'm not sure this would also be a good strategy for multiple system atrophy, since the pathology is so different there, and because you also have a lot of striatal neuronal loss. But I think I should ask this question to probably the both of you, because I think you'll also have a good answer to that as well.
So if I was going to answer, go ahead, Patrik.
Yeah, go ahead, Jeff.
So given the lack of other therapeutic avenues right now, I think trying stem cell transplants in a patient with MSA is not an unreasonable path going forward. We know that we can get striatal cells to survive in animal models. We can get nigral cells to survive in animal models and in patients with Parkinson's disease. We know that the safety profile is pretty good. And so I think that it's not an unreasonable approach to replace these neurons with stem cells. Patrik.
Yeah, so I'd echo that, but I would also want to emphasize that it might be mostly for the MSAP type of disease, where the basal ganglia are primarily affected. And there were several studies about 20, 25 years ago in animal models of MSA, where in particular the group from Innsbruck in Austria transplanted cells to, I think, as you were suggesting, both dopamine neurons and striatal neurons mixed. I would also argue that it would be a challenge to reconstruct the circuitry because of two different populations being affected. And I would also say that MSAC, it would be hard to know what cell type to transplant and exactly where. So MSAP, I think, yes, it might be worth trying because of the lack of other therapies.
Would you put the nigral stem cells in striatum and the striatal stem cells in nigra? Would that be an easier path going forward?
Yeah, that's an interesting concept. I think the group with Gregor Venning, Werner Perver, Nadia Stefanova, who did this initial work, if I remember 20, 25 years ago, I think they mixed cells and put them in the striatum. This is my recollection.
So Dr. Van Leer.
Only in the striatum.
Okay. Dr. Van Leer, let me just ask a quick question of you. So the gene delivery of GDNF has had a bit of a checkered paths for Parkinson's disease. What gives you optimism going forward for MSA?
So I just wanted to add, there is a mesenchymal stem cell study that's being done. It's been looked at in MSA. Just want to throw that out there without the direct delivery. It's
mesenchymal stem cells don't
want to
survive.
throw it out there.
That's
throwing it out there. So and then on top of that, GDNF, though it may not restore, bring back cells that are dead, but there's potential, we're hoping, that actually to maintain the health of the cells that are still there, maybe sick by, the processes that are causing MSA, but potentially restoring them back to health. So not direct replacement, but restoring that way. In regards to our gene therapy, were you suggesting that as a different approach for, can you, I was wondering if you could repeat your question.
So Dr. Holec, as a person who does neuropathology on human brain samples, it's been asked whether MSA proteins, which have a prion-like property, are toxic to researchers and another
You are froze, Jeff, for me anyway, so I'm not sure if there was more to the end of that question, but I'll answer what I heard. So there's no data that suggests that any misfolded alpha synuclein is dangerous to researchers themselves, which is true for PRP prions like Kuru and Creutzfeldt-Jakob disease. So there's no data that suggests that it's dangerous to researchers, but out of an abundance of caution, our lab treats everything like it could potentially be dangerous, just because it's a good rule of thumb when you work with any disease is to always treat it like it could harm you. So even though there is no danger of it to us that we know of, we treat it as if there could be. I also saw a question pop up in terms of if it's dangerous, about like if people could give it to their family, and that's kind of in the same vein of this question, which is along with what I mentioned about Kuru being able to be transmitted between people, or Creutzfeldt-Jakob disease, getting that from eating infected meat. Like, that's not something that we've ever seen happen with synucleinopathies. So that's not anything that anybody needs to worry about, giving it to a family member. This is more of just a caution for researchers. It's always better to be safe than sorry.
I mean, I will tell you that Stan Prusner, a Nobel laureate, believes that we should take extensive precautions handling MSA tissue, because he believes it is a, quote, prion. But I agree with your answer. So Dr. Gan-Or, a couple of questions. Is there precision medicine involving genetics happening now for MSA? And also, if you could follow that up with, how do you figure out if people have a GBA mutation? Is it only after death, or can that be found out during life?
Thank you for the question. So feel free to correct me if I'm wrong, other members of the panel, but I don't know of any current study that is targeting specific subpopulation in the sense of, from the point of view of genetics, which would what we mean by precision medicine. So, for example, targeting specifically people with GBA mutation. So I'm not aware of any trial like that, but if there is one, feel free to comment about this. As how to find if people have GBA mutations, we don't have to wait after that. We can find it in blood of patients, so people who contribute blood or skin or any other sample basically that can be tested for DNA, we can test for the presence of GBA mutations. Having said that, just to comment that the association between GBA mutations and MSA is really not clear. I talked about this in my talk. There is some evidence that support it. There is some evidence that do not support it. And it's still unclear whether GBA mutations are really contributing to MSA.
Dr. Norcliffe-Kaufmann, why is it so difficult at times to diagnose MSA?
That's a big question, and it's an excellent question. I think the difficulty arises because MSA can have other diseases that mimic it. And then you have diagnostic criteria where we have to wait a certain amount of time in the clinic for people to have developed a sufficient amount of typical symptoms to be able to diagnose MSA. right? So you have this period of time where in clinic where you see patients, you may suspect that they have early MSA, but it isn't until the point where they've lost a sufficient amount of neurons and they have, the core features of the disease that we can make a clinical diagnosis. And then one of the things we know, the diagnosis is made at pathology, right? So by detecting the pathology that we're all talking about right now with these abnormal synuclein deposits. So that's the definitive diagnosis, but we have these clinical stages. And then there can be other diseases that mimic MSA. So as we're, sort of seeing patients in clinic and collectively, putting our thoughts together, we're becoming better able to identify those earlier stages of MSA and come up with sort of criteria to be able to diagnose it and pick it up in earlier stages.
So let me follow up. Most, and we talked about this early on in the discussion, MSA is such an aggressive disease, but there are some patients who have very slowly progressive disease. Do we have any understanding as to why that population progresses slowly versus others are so aggressive?
I mean, first of all, that's absolutely right. And it's one key observation that we see. I mean, when patients are given the diagnosis in clinic, there's often a temptation to look at, average survival and things like that. But as we've tracked MSA patients over time, we do understand there's a group of people that progress very, very slowly, right? And at pathology, they do have, a definitive diagnosis. So, that's it. It becomes very interesting to follow those patients to understand what it is that is unique about them that causes the process to occur slower and, of course, be associated with longer survival. As to what's happening at a molecular level, I don't think there's any better people to answer that than others on this call, right? So I wonder if there's anybody else that has ideas about what could be going on at a molecular level.
Yeah, I could add something here in the discussion. It's a very interesting and important question. And I think that what we need to say is essentially that we are not all alike. We do not have exactly the same genes and alleles. And it is through these combination of alleles that some will evolve and develop, unfortunately, the disease fast, while others will develop it slower. We are not equal. That's what I'm trying to say.
You're not equal. Go ahead, Patrik.
So Ronald, I'm surprised you didn't say that we're also not equal when it comes to the shape and form of those protein aggregates. Oh, there was a cat just went across Sarah's screen. I don't know if you all saw it, a big black cat. Scary. Anyway, so
you are right.
So you talk about spaghetti linguine and macaroni type of shape aggregates. Do you think the initial shape of the aggregate could govern the speed of progression in some way? Forget about the genes just for 20 seconds.
Yeah, thank you, Patrik. Definitely, and you are perfectly right. But what defines the shape of these pasta are the molecular chaperones, are our alleles that we are expressing to different extents.
So let me follow up, Dr. Melki. So we've shown that patients with one cardinal sign of Parkinson's disease have rampant synucleinopathies. Do you think MSA is the same? That by the time we get a diagnosis, that the aggregates are already formed and we have to use that information to guide our therapeutic interventions?
Definitely, Jeff. So the thing what is missing here are diagnostic tools. So tools that can distinguish one polymorph from another to sort of predict how things are going to evolve, because we have been able to generate many polymorphs, as you know, including from patients. At the resolution we have the impression that the polymorphs coming from different MSA patients sort of look alike, but we might discover that they are still different to some extent. But what we are missing are ligands that distinguish one polymorph from another. We can obviously not do like a cryo-electron microscopy structure from every single patient, including from biopsies from the periphery.
Dr. Peelaerts, what is your level of optimism about finding a biomarker for MSA? And where would that biomarker be? Would it be an imaging biomarker, or CSF, blood? What do you think going forward? And do you think that that is one of our major unmet needs in terms of understanding MSA and finding a better way for therapeutics?
Yeah, absolutely, absolutely, and I think there my optimism is actually pretty high, and I think this also relates to some of the things that Ronald was just mentioning, and I think there has been some I think very exciting studies that have shown that even just with small samples of CSF, you can actually detect some of those, specific aggregated proteins in, for instance, spinal cord CSF samples from people with multiple system atrophy, and it even allows to differentiate between people with multiple system atrophy and people with Parkinson's disease. So I think this is a very exciting field, and I think this is a very promising field. So my level of optimism is really high there.
Thank you, Dr. Henderson. The microbiome is getting a lot of play in Parkinson's disease. Could you discuss a bit whether you think the microbiome plays a role in MSA? And if so, do you think that taking prebiotics or probiotics would be a safe, easy therapeutic intervention for patients with MSA?
I think maybe this would be best directed to Patrik and Wouter. I think that this is their area of research, whether infectious diseases or, in this case, maybe microbiome differences in individuals could somehow trigger or predispose this disease. I think I would leave them to answer that.
All right, Patrik.
And I was so relieved that you got the question, Mike, because, of course, I don't really know what the answer is, but I can speculate. And Wouter, you're welcome to chip in. Anything that can modulate inflammation in the gut, and we talked about the rectum, might be relevant for MSA-C. And I said that I think it's about 30% of people with inflammatory bowel disease have inflammations of the lowest part of the gut. So anything that can modulate that, you could potentially imagine plays a role. And there are some studies on microbiome and MSA, but it's an emerging, evolving field, and I don't think there's anything definitive one can say. It's very complicated because there's so many different types of bacteria in the microbiome. Some seem to promote inflammation, some seem to reduce inflammation. What do you say, Wouter?
I agree, and I think it's a very interesting, let's say, proud or it's an interesting idea where different infectious triggers might either influence or even act as a cause or as a pure trigger of multiple system atrophy. If we just, for instance, look at genetics, it's been very hard just to identify just risk factors or genes that might be associated or causative of multiple system atrophy. So clearly something else has to be happening there. And so we've, of course, been using the UTI model just as a model to study how the disease might start and just to have this as a sort of paradigm. But I think there could be other challenges or infections, and as to that, I'm just, for instance, thinking about certain even coronaviruses that can specifically infect, for instance, oligodendrocytes through the facial nerve. This has also been described, and this has been a popular hypothesis in multiple sclerosis, where you also have a lot of demyelination and several of the pathologies that actually overlap with that of multiple system atrophy. So I think it's an interesting hypothesis, and also certainly for my future research, this is something I would like to dig into more. So that's like I said, I think it's an interesting avenue, but there's absolutely not much known about it at this point.
Dr. Holec, if I could give you $1 million for one grant, and you could have a choice of 3 different avenues to study, would it be misfolded proteins, autophagy, or inflammation?
I don't know. I do love my misfolded protein. I love them very much. There's still a lot to learn, I think, about how one protein can adopt so many conformations. Then try as we may, I feel like we've barely touched the tip of that iceberg.
Okay, I actually would choose inflammation or autophagy because then you have all the co-pathologies that you can address. So we have time for just one more question. I'm going to leave it to Dr. Van Leer. There's a person in the audience who wants to know, is gene therapy dangerous?
That's a really good question. And if you're not thinking about that, I always make sure that it gets brought up because it's important. So, just like anytime we do brain surgery, there are risks that are involved. And though I don't think that this is minimally invasive as compared to other types of brain surgeries, this is minimal. I think we get into more trouble with brain surgery when you're actually taking a lot of stuff out, and that's not at all what we're doing. And we're also not leaving anything behind, and that's another source of problems for other types of interventions for neurosurgery. That being said, it's still, we're putting something in the brain, and anytime you're putting anything in the brain, there's always going to be a risk of things like bleeding and infection. We haven't seen any types of infections that have occurred, but there have been cases where there have been small bleeds in a few patients. These are in other studies, not in ours. But that being said, they've been small, and if they were symptomatic, they were mild and eventually resolved over time. The risks are not much different than things like deep brain stimulation. So just to keep that in mind, this is an FDA-approved therapy where, again, you're implanting electrodes, and they then stay inside the brain for long periods of time. So it's very similar to that. So it's not without risk by no means, but for a lot of people, sometimes the risk is worth the effort of going through these types of studies.
Well, thank you very much. Well, that brings us to the close of our session. I want to thank all my dear friends and experts in this area for their time and their insight. And with that, I will draw the session to a close. Thank you very much.
Thank you, Jeff.
Thank you.